and it is my pleasure to introduce our speaker today, David Spotts. David is a biotech executive with experience working on ataxia drug development. He also serves on the National Ataxia Foundation's board of directors. He has seen the impact of this disease on families affected by ataxia. And in his career, David has worked closely with patient advocacy groups from an industry perspective. He's also very excited for the opportunity to be on the board of the National Ataxia Foundation to bring his experience and support an organization with such an important mission. So with that, David, the floor is yours.
Awesome. Thank you, Celeste, and super excited to be here with you guys today. Love seeing the wide-ranging audience, and hopefully this is a useful time. I thank you for taking a little time and listening to me, and at the end we'll have tons of time for questions, and hopefully you can find some relevant information for you today. If this is your first time hearing about drug development, totally appropriate. If this is if you're an expert on drug development, hopefully you find something useful out of today, and also welcome a discussion. So hopefully this will work for a wide range of perspectives and backgrounds from folks. I'm hailing right now calling from Boston, Massachusetts, where it is a little bit chilly. The fall has apparently disappeared on us, but originally from Georgia. So excited to see some folks from Georgia on the line, and have been a part a little bit recently of the board of the NAF, and just found the work super fulfilling. Excited to get back. As Celeste mentioned, I've worked in drug development for ataxia, and know it's a space with a huge amount of unmet need. And I think from today, really what I want you guys to hear is a little bit on what is drug development, what are some key terms. Hopefully this is not too remedial for everybody, but really hopefully to learn a little bit and serve as a foundation for folks moving ahead with drug development. A lot of exciting stuff in the ataxia space. But I'll start out with what's my why. So this is a picture that I keep of my desk of two photos, three photos. On the left and right are my daughters Nora and Tess, who are a little older now, as you'll see in a sec. But this was pictures of them when they were sleeping and peacefully quiet when they were born. But in the middle there is a picture of a gentleman in a wheelchair, and on his lap is my grandmother, my maternal grandmother, and the gentleman was her brother. He had, well, we think he passed away before kind of some of the health diagnostics were available, but we think he had Duchenne muscular dystrophy, which I guess does run my family. And so for me, that's always been an important why of understanding that the genetic diseases run in families often. And that's very much a reason why I have kind of gravitated towards the medical field and working in pharmaceuticals and biotech, is really to give back to families, possibly like mine, who have similar things in their family, are affected by conditions, or living with conditions. So if I can do my small part to help, improve other people's lives, I maybe sleep a little better at night, and excited to help the world be a little bit better place. So that's my why. And here's a few more updated pictures. On the left is our family back when it was a sunnier fall, a little warmer day. Nora and Tess there, Tess on the left in the bluey outfit, Nora in the right on a ladybug, a French cartoon on Netflix, if you haven't seen it, if your kids watch it. So that's them pretty recently. And then on the right there is my day job, where a company I work for develops treatments for a variety of diseases. One of them is macular degeneration. So you may have seen some ads our company has on TV with Henry Winkler, just bringing awareness to the disease, just in case you've seen those. Apologies for the commercials. But what I hope you get out of today, so an increased understanding of how drug development works. Again, kind of no matter where you are in your learning journey, I hope you learn a little bit more today. I hope you get a little bit of knowledge around terms and language used in drug development. Some of the insider lingo, some of the things that folks like me and others just happen to say and don't even think about what they mean sometimes. Making this presentation was very helpful for me to go back and be like, why do I say that word and not this word? So let's talk a little bit about some terms. But I think importantly, a view from somebody in the pharmaceutical industry, and maybe like you, I'm not an MD, I'm not a PhD. I approach things from just a very common perspective and try to understand things. And so maybe that's a useful perspective for you guys, and maybe some of you are going through that same process of trying to learn and understand how this all works, because it is complicated. But I hope I can simplify it a little bit today. A few important points. All opinions are my own. I'm not speaking on behalf of my company, or I am on the board for the NAF, but these are all my opinions. None of this information should be taken as specific medical advice for you or someone in your family or a friend. And for any specific medical questions, please consult with your healthcare provider, your doctor, to make sure you get the information that's right for you. Questions around specifics around, a drug or a treatment, always best to talk to your own doctor on those things. And I'll mostly be talking in generalities today, but certainly on some aspects specific to the ataxia community. So to start, I love to start a little bit of history. So where did the pharmaceutical industry start? Well, I think really for much of human history, medical treatments, medicines, drugs were from natural sources. Plants, herbs, even clays have a medicinal property. People didn't exactly know how they were working, but they knew they seemed to have an effect. Even the word drug comes from the barrels that the herbs were stored in. So there's even a connection of the word back to that herbal approach. But I think kind of the real kind of pharmaceutical industry started in the 1800s. Many of you, I'm sure, have heard of the Industrial Revolution that was happening around this time. And scientists were able to start purifying chemicals, make things in larger scale or larger amounts, so that if they had a discovery, they could make it for a lot of people. Many of the companies that you've probably heard of, Pfizer, Merck, and many of these companies, they had their starts in this era. The first placebo-controlled study, which we'll talk about what a placebo is, was conducted way back in 1863. That's that scientific method taking hold in medicine. A lot of the initial work was focused on painkillers. That was certainly a huge topic of the day, is how do we, make sure people are in pain? How do we relieve some of that pain? But I think two of the significant breakthroughs that signal the start of what I call maybe the modern era of medicine, insulin and penicillin. Both of these around the 1920s. You can see there on the right a picture of Dr. Alexander Fleming, who famously discovered penicillin, and the mold near the window was grown. The mold stopped growing or the bacteria stopped growing with the mold on it. So I think that this is kind of the start of the modern era of really new medicines that really could change people's lives and affect and slow down diseases or prevent diseases. So, what is a pharmaceutical? In some ways, it's a fancy word for drug. If you go to dictionary.com, it's just a chemical substance used in the treatment, cure, prevention, or diagnosis of disease, or used to otherwise enhance physical or mental well-being. That's a long definition. If you go to the FDA, what they would say, it's the primary intended use of the product is achieved through chemical action or being metabolized by the body. The product may be considered a drug. I added the underlines, not from the FDA. So what that means to me is that it's not a device. It's not something that works through mechanical means. It's not something just purely for, it's not like an imaging tool for diagnosing or looking at affected MRI for the brain, for example. It's something that works through chemical. It's something that typically we'll talk about how you might take a drug and different types. But the way I really think about it from the insider perspective is the benefit risk ratio. Is the benefits of this approach outweigh the risk? To me, that's a medicine. If there's no benefit, it's not a medicine, right? Why would you take that? And often many drugs do come with side effects or adverse events, which we'll talk about. And it's important as in the drug development process that we're constantly thinking about this. Will the benefits of this approach, this medicine, outweigh the risk? Will this be appropriate for people with a disease, with a condition, at different stages of that condition? And we'll talk a little bit about that. But to me, that's really what is a pharmaceutical. Again, a little bit of a fancy word for drug. But again, it's something that has benefit to somebody living with a disease and may be associated with some risks. So how do I think about describing a drug? And many of these you're probably very familiar with. You could think about the way in which you take it or the route of administration. And that can be oral, a pill form, a liquid form. I'm sure you guys have seen these drugs. These are taken. I didn't get permission from Johnson & Johnson on these, so apologies. But topical administration, drops, hydrocortisone, lotions, right? Very, very common approach. A little bit maybe less common, injection, subcutaneous injections or intramuscular injections or intravenous injections. Certainly, if folks have been to the hospital, you may have gotten an IV. And I have a typo there. Intrathecal, typically I should be IT, sorry. That is a kind of I think increasingly, it's not a common approach, but in some fields, some ataxias, certainly that is an area of research of administering a drug directly into the to the CNS area through the kind of the lower back area through an injection. Sounds painful. It is a relatively established procedure with certainly some risk, but a relatively established procedure that is increasingly being used as a way to get medicines directly to the tissue, which in that case, the brain or the spinal cord, where you want to get the drug to. So in the drug development world, we think a lot about how are we going to get the drug to the tissue of interest, to the area of the body we want to treat. And each of these routes has advantages and disadvantages. Oral, very, very typically pretty easy to take a pill for the most part, assuming they're not those giant pills. But maybe harder to get the drug to the tissue because it's got to go throughout the body. Intrathecal, a little more invasive, needs to be delivered under very certain circumstances by a trained specialist, but maybe able to get the drug better to the area of the body where we want. So that's kind of one way we think about drugs. We also think about the type of the drug, or kind of a little bit how it's made. So what we call a small molecule. This is, I'm not a chemist, but this is, just a chemical compound. This is actually the chemical compound for acetaminophen or Tylenol. So that's going to just made through chemical means. You think of a scientist with beakers in a lab. That's how you would make something like a small molecule. A little bit easier to make, pretty small by its name. Then there's a little bit more of a newer approach, which is a biologic or a protein therapy. What's depicted here is a medical antibody, Herceptin, that's used to treat breast cancer. So this is a more modern approach, making antibodies, humanized antibodies typically made through a cell line that it can be used to treat disease. And that can be a very useful approach because it can latch on to certain parts, certain things that shouldn't be in the body, or cancer or other ways. So that's a little more modern approach. A little bit, certainly much bigger. We often call these large molecules or biologics. They're bigger. They're made in a certain fashion. They're much bigger than a small molecule. And then even more modern approaches. Folks on the call are probably familiar with viral or non-viral gene therapy, where a little bit of some genetic information is inside a kind of a viral virus outside that is delivered to hopefully lead to maybe a little more permanent change in the course of the disease. And that can be in a virus or again non-viral, where it's kind of a little bit of a membrane around that information. And those approaches there are some approved gene therapies in other areas of medicine. So that is an area that's certainly of active interest. And then cell therapy. And when I'm thinking cell therapy here, I'm thinking delivery of certain CAR T, certain treatments are delivered by either taking cells out of your body and modifying them and getting back to you, or deriving some approach. I'm not really thinking stem cells, if you've heard that term. I'm sure there's some information on the NAF website about this. But I'm thinking more cell therapies that have gone through a clinical development process. And each of these methods, again, has their own advantages and disadvantages. And from a drug development perspective, we're thinking, can we go small molecule? Can we go with the protein approach? Can we go with a more permanent, hopefully more durable gene therapy approach or something? And each of these has risks and to the way can it show the effect that we want, different safety risks. So this is one way we're thinking about a drug as folks are designing it. But there's a lot of other ways. We think about the outcome. And we'll talk about outcomes a little bit, but the outcome we're hoping to achieve. We're trying to modify symptoms. Painkiller, very common example of modifying symptoms, just to relieve the symptoms. Or maybe better, do we want to modify the course of the disease? Do we want to slow down a progressive disease, or so hopefully halt a progressive disease, like some ataxias? Or preventative, do we want to prevent anything from ever happening? And that could be through a vaccine or through other approaches. We think of it by the mechanism of action. That's just how the drug, we think it works. And I italicize think in humans, because often we don't exactly know how it works in humans. And that may sound scary, but the reason we say that is we can't always exactly get the tissue. If it was brain tissue, we don't want to be doing brain biopsies frequently, right? That's not a great thing. So we do a lot of animal testing in other ways, and we think we have a good idea how it works in humans, but we never know for certain. But in the end, if it has the effect that we want with a good safety profile, maybe that's okay. We don't exactly know how it works. It's more importantly that it does work. You may have heard the term drug class. These are compounds that have maybe a similar mechanism, a similar way of working, maybe a similar structure, treat similar diseases. And the reason we often think about classes, they may have similar properties. If one drug in a class has a certain side effect, other drugs in that class may have that same side effect or adverse event. But it's not 100%. And that's why you guys probably know there's multiple kinds of antibiotics, multiple kinds of steroids, and there are classes of antibiotics and classes of steroids, and the properties of each drug may be a little bit different, but it often can be a way to think about them in general. But this is where it gets really nuanced, and why you need your doctor working with a pharmacist very closely and the full healthcare system to make sure we fully understand how the drug is working. So how does a drug come to exist? When I think about it, this is a very, can be a long process, can be a short process, depends on what we're looking at. But it all goes back to the scientific principles or the foundations of what we're trying to understand. That's that discovery and preclinical work that can be in a dish, in a petri dish, that can be on increasingly on a computer, understanding maybe the genetics. And what I think about at that stage is that's research. And you guys, I'm sure, heard the term R&D, research and development. Research for me is that discovery and preclinical stage. Then it goes into humans, clinical trials. To me, that's the development program. That piece can range in the number of people, and there's different phases that we'll talk about. But between those two stages, the discovery, the preclinical work, and the clinical trials, there's what's called an IND. That's when you have an investigational new drug. In the U.S., that's a lot of paperwork you file with the FDA before you can start testing in people, to make sure that all of your work supports that this is, we think that has an appropriate safety profile, that we think that the adverse events we may see from the drug are reasonable for the condition we're hoping to treat. Also that there's evidence typically in some animal species that there is a drug effect or it's reaching the target. But that the FDA says, yes, we based on your information, this is a drug, an investigational new drug that we can start testing in people in a clinical development program. In my mind, it's not a drug. It's an important point. Not a drug until the very end. Here, it's an investigational new drug, an experimental drug, but it's not a drug. It's not a medicine until the end. Then you go through clinical trials. Lots of terminology we'll talk about there. That's that development phase, and at the end of that, you submit an NDA, a new drug application, to a regulatory body. In the U.S., as I said, that's the FDA, but that could be the EMA in Europe, the European Medicines Agency, or Health Canada. There's groups like this all around the world for every country. You submit for regulatory review. So they look at all of the information. That NDA, that application, it used to, I'm not kidding, in some cases would take up like all of the clinical trial files would take up like a tractor trailer full of paperwork. Now, luckily, it's digital. It does take a long time to transfer that file over, as you can imagine, but it's a lot of information. All the clinical study reports, all of the information that was collected in the clinical trial, but also in any animal testing that was done, to make sure that the regulatory body, again, probably the FDA here, can look at that and say, yep, this is appropriate. We can understand it. We have all our experts review it. And at the end of that process, it can be up to a year or longer, depends on what the FDA does, is that FDA approval or the other approval by a different group in a different country. And that's, for me, that's a huge milestone for everybody in the drug development. That's a huge milestone for many people living with conditions. That's a huge milestone, right? That signals that there is now a medicine, a treatment for a disease. It's been characterized. We have the information. Information can start going out to the world about this new medicine. And that's what starts a lot of what I do in the kind of commercialization world of educating folks on that treatment, but also post-marketing surveillance. The kind of learning about the treatment doesn't stop just because the drug is approved. It's not like the FDA says, here's the drug, go out and we never hear from you again. There's still surveillance. We're still watching for, are there adverse events that we didn't pick up in the clinical trial process because we were testing a certain group of people. And in the real world, the real world has real issues, right? And we learn about real things. So we still watch the drug very closely in the real world to see how it's being used, and also to do more studies to say, maybe we can look at longer time between injections, for example, if it's an injectable drug. We're constantly learning in the pharmaceutical industry to make sure we're providing the best possible medicines for people living with these conditions. So as an example, there's my animation. As an example, for Skyclaris, I'm not speaking on behalf of Reata or Biogen, but just as an example from publicly available information, their IND, their investigational new drug, was submitted in 2014. They went through their clinical development program, their clinical trials, submitted their NDA, or the NDA was accepted on May 26, 2022. So it can take some time to go through those studies. Their approval a little bit under a year later, in February 28th of this year, huge development for the field for this therapy. So you can see it does take some time, and I didn't only have the date on here of when that basic research started. It could have been years and years before, where really the first inklings of this approach were started to work its way all the way through. So I don't have that full timeline. Not trying to intimidate anyone with the process, but just to show that each stage is very thoughtful and carefully designed to make sure we're getting the right information at the right time. So a few concepts in key lingo. One concept, so what's the purpose of a clinical study? Is it to pass or fail? To say if a drug passes or fails? It's not the way I like to look at clinical studies. I like to think clinical studies answer a question. They ask a question like, will this approach, this chemical, this molecule in this disease, in this patient population, with this measure or this kind of evaluation, show an effect? And that effect could show a benefit on slowing disease, could show a benefit on safety. And the answer might be no. And that can be, yes, I recognize that's a very tough answer sometimes to hear, that it didn't work, but that moves science forward. There's a lot of work done on how many molecules actually make it through for every how many don't. And that's how science works. And that's how we get to a safe and effective medicines, is by testing things and asking these questions. So again, I don't think as you see results, it's certainly great if we see positive results. It is also okay if we see results that we don't think are positive, because it answered an important question. And that the answer may be that just in that part of the disease, in that stage of the disease, it doesn't work. But maybe that means it could work in a different stage of disease, or for a different population, or with a different measurement method. So that's why I don't like to think of it as pass-fail, but again, answering a question. And that question is often related to what you may have heard of inclusion-exclusion criteria, which are just ways to say we want to look at a certain group of people living with a condition for a study. It's not to make it, I know of examples where folks are like, well, I'm 49 and the cutoff is 50. Why I'm so close to 50? Why can't I, and I won't go into all that now, but I think it just at a high level, think of inclusion-exclusion criteria as a way to define the groups very carefully so that we can get an answer to the question. Because if we don't think of it in a very careful manner, it's very hard to get that answer. And that to me is really the only bad outcome of a clinical study, if you don't get an answer. If you don't even get a yes/no answer, did the drug work or didn't it work in that population, that's a bad outcome. If you don't have defined inclusion-exclusion criteria and carefully control them between the group on the drug and the group maybe on a placebo, that's why we have those. And that's that control arm, which is an important point. Many of you, I'm sure, have all heard of a placebo. Often if you take if it's an oral medication, you're taking the active drug, that what we hope is the active drug, in comparing to something that's not the drug. And our hope in designing programs that are placebo controlled is that the person that's taking what could be the medicine doesn't know if they're getting the placebo or not, so that they can't change anything in their life to say, well, I'm on the drug, so I need to eat healthier. We don't want to bias the study or affect those results like that. And also the person even administering it. That's why we say a double blind. The person providing the drug doesn't know if it's the sham or the placebo or the treatment, and the patient doesn't either. That's that double blind. Or you can have studies that compare to an active comparator. If there is a treatment already approved, you don't want to sometimes compare to a placebo, because that would be taking somebody off of that active group or an effective treatment. You maybe come to say, is the new drug better than what's on the market already? It's typically a harder study to do, but that's the kind of thing that pharma we should be doing to show that we're not just comparing to nothing. We should be comparing to hopefully something that works. That is kind of the gold standard. You may have heard that term: randomized, double-blind, placebo-controlled studies. Randomized, that just means that we take everybody and kind of randomize them into two groups to make sure that the groups are very similar between maybe the placebo and the treatment group, or what we hope is the treatment. Double blind, I talked about. That's again, the patient and the person administering the medicine doesn't really, or evaluating the patient doesn't know what they're on. Placebo control, again, having something that takes out the bias to make sure that they don't know it's a placebo that often almost like a sugar pill. So that's what you may hear as the gold standard. Another term you may hear of, pipeline. It's just a way to shorthand to say it's those drugs that are kind of coming down the pipe, investigational drugs or experimental compounds that are coming down there in development that may be approved. And it's an important thing to think about how long those things in the pipeline will take, what's in the pipeline. We want to, in the Ataxia field, we want to have it's good to have a lot of drugs in the pipeline. I'd love to see that there every day. I see a lot of exciting developments in the Ataxia space that for me give me a lot of confidence that there's a lot of stuff in the pipeline, and I'm really excited about the future. So to break up the talk a little bit, I had a little quiz in here. So if you just drop in answers into the chat, Celeste is going to help me out and see if anybody's gotten the right answer. So some of these I've talked about, some of these I haven't. So we'll see where folks know. So one of the terms that you may hear in the field is they're seeing written out FIH. I'm curious if anybody knows what that means. Celeste, tell me if you see any answers coming in.
Will do. So if anyone has any ideas of what FIH stands for, you can put your best guess in the chat and I'll read them out to folks.
This is my test to see if anybody from the drug development industry is on here, double checking me.
And if you don't know, that's okay as well. We'll give it another 30 seconds. Oh,
I hit the
there we
button
go.
too soon.
That's okay. We had someone who put first in hand. Oh, we have first in human. And the answer was first in human.
And this is an important milestone, right? That this signals again that step from the preclinical research into development. When drugs go at this stage, the first patient that's given the drugs watched very closely, sometimes admitted to the hospital overnight, just to make sure we can monitor their vital signs, their heart rate, their blood pressure, other things, to make sure that, hey, let's watch very closely to this drug. This is the first time we've ever used it in a human being. Let's make sure it's safe, right? And often early studies are watching just for safety. The next term, which I did talk about a second ago, see how good I did explaining it. IND. Any answers to IND?
Now we already have some folks quick on the draw putting things in the chat. We have someone who put investigational new drug.
Right, that is correct. And that's that same, when you go first in human, that's an investigational new drug. And keyword for me to have that investigational. It's not a drug yet, not just a drug, it's an investigational drug. We're looking to see if it could be a medicine, a drug. All right, an NDA.
So we have someone who put non-disclosure agreement, which is what I thought it stands for when I first started in this my grad studies. I thought it was non-disclosure agreement, but we have a few folks putting new drug application in the chat.
And the reason and that's exactly right. And for this talk, at least, maybe the non-disclosure agreement for a different talk. The reason I think it's good to think about these is I mix these up all the time. If you do that, have no fear. It's totally the letters are very similar. I think maybe I before N, investigational is the start. New drug application is towards the end. I think that application is what we give to, say, the FDA to say, hey, we think we have a drug here. We've done a lot of work. Can you look at it and tell us, what you think, FDA, to make sure that hopefully we can get that medicine onto the market. PI also has a couple meetings, so I'm curious if people.
All right, people are putting some questions in, and so in the UK, someone is sharing that it means patient involvement. Some people are saying patient input. Some people are saying PI 3.1416, principal investigator, which is where my mind goes to, or primary investigator. But what does it mean in this context about drug development in the US?
Yeah, so I think of it in terms of prescribing information. For a medicine that you guys, I'm sure many of you have in your house, if you go, often there would be a little pieces of paper folded up, very small, lots of folds, that has the prescribing information. This is the information that in this case, the FDA says is very important to share with the world, or share with the US, especially for healthcare providers to look at, to have information about the safety of the drug and the adverse events that have been seen, any warnings and precautions to think about when possibly using this drug, how the drug is administered, how the drug is thought to work. Also the clinical study data. It's kind of the summation of everything that we've done, excuse me, up to this point to get this drug approved. And it's a very important document. I know that I'm sure many of you say, oh, it's that piece of paper, throw it away. But if you have a chance, take one out and look at it, and you'll see some interesting information in there. Often there is a section and they're about patient counseling. There can be some other documents provided that are summary documents for patients to read. But it is again, it's that culmination, that summation of all the stuff that's gone in. Say this is the very important information about, as you think about prescribing this drug. It's the primary audience is that healthcare provider. But I think it's a neat thing for folks to know that on our side, it's something we work towards very carefully to make sure that just the right information is out there. It can also mean many of those other things that you guys put in there, but for this talk, it's the prescribing information. All right, so how do we think about clinical trial programs? And when I say a program, I'm just thinking about all of the different stages of the trials that a drug might go through. And it's important to note that clinical trials can vary, right, based on the disease we're looking at, the prevalence, how common is the disease, the severity of the disease, the how fast is progression, how fast is the disease, what are the measures that we can use which you talk about, the outcomes, that to evaluate, and importantly, what question we're asking at each stage. So often this is kind of the traditional approach. I will say it's gotten very complicated in a good way over the last few years, and there's a lot of new cool approaches to try to get things going faster, but I'll start with this. This is kind of the traditional approach. And often the first stage is that phase 1 safety. That might be 15 to 50 patients. It might be a smaller number, but not that much larger. And really often the key question at this stage is safety. This might be done in people without the disease, volunteers, and at often a very low dose. Say, all right, does the drug seem to be safe? And how does it interact with the human body? We're not really worried about efficacy or the effect of the drug. Efficacy is just a fancy word for effect, in my view. We're just looking at safety. And we want to do it in the minimum number of people because we don't want to, if the drug isn't safe, if that's the answer to that question, if it's not safe, we don't want to have exposed, given that the drug to a lot of people, or the possible drug to a lot of people, and hurt them. So we test in the smallest number of people possible. And it's not typically that long of a study. Sometimes it's just one dose to start. And then we add in more doses, or one very low dose, and then we increase the amount. But that kind of takes us into phase 2s, which are often safety and dosing, or safety and hints of efficacy. More patients, right? And maybe a little longer. So we can start to see, all right, in a larger population, seems like it was safe in the phase 1. Now let's look at a few more people. Is it still safe? Is it safe at what we think is maybe the closer to the right dose? Can we get to what we think is going to be an effective dose and still have a good, not have too many adverse events or side effects, have a good safety profile? We're watching closely for side effects here. We start to have a pretty good idea of what we might see. Importantly, we want to say what is the best dose? Where do we get the most benefit for an acceptable safety profile? And we're also starting to check for effectiveness. It's not the primary reason to run these studies of phase 2, but we do want to say, does the drug seem to be having an effect? And is the effect such that we think we want to go into a larger phase 3 study? Sometimes you need, in some indications, some diseases, you need 2 phase 3 studies. So you want to say, hey, those are expensive, which we'll talk about. Does the effect that we saw in the phase 2 with the safety that we saw in phase 2, is it appropriate where we think we can now go into phase 3s? And there are FDA meetings or other regulatory body meetings throughout this process that we're working in collaboration to make sure that we're answering the right questions, asking the right questions, and getting answers that help move us on, hopefully getting to a medicine. Phase 3 studies are more people, but again, it depends on the disease that we're looking at. In heart disease, you could have many, many people. In a rare disease, it could be a much smaller population, because in for example, some ataxias, you're not going to find thousands of people. And don't worry, if you go online and you look at diagrams like this, it's very easy. I've done it. It's very easy to find examples that you might say, what, that says 1,000 people. And that might be, though, an example for a heart disease, cardiovascular disease, diabetes, things that affect lots of folks. But for rare diseases, the FDA, is, I think, especially, and many other of these groups, are very aware of the population sizes and trying to think, with industry very creatively about how do we design clinical trial programs that can get to an answer on the effectiveness and safety of a drug in a much smaller number of folks. And And so I think that's kind of the traditional framework, but there are lots of cool examples out there and ways to think. Again, how do you make it shorter? How do you make it, what's the smallest you can make the study with the least number of people but to be confident, because that's where that's where the trick is, to make sure that you're confident in this group, because after that FDA review, right, that's going to start going out to more people. Are we confident that this drug will have a similar, or hopefully the same, maybe better effect in the real world, and importantly, a safety profile? There are examples, unfortunately, where things have made it past FDA review, but when used in the real world, new safety events happened. Vioxx is a classic example where there were drugs that have been pulled from the market. That's why I say it's not totally done at FDA review, but we do want to make sure that at that point that we're confident that as the drug goes out into the real world, that it can work and it can hopefully be safe. But again, each disease is treated differently. Each molecule, the way the molecule works also can affect this. So a lot of differences here. So this is not one size fits all, but it's kind of a good starting example. So one term you may have heard, endpoints, which is kind of a historical term. Honestly, it's more about how studies used to be done. I like the term outcome measures. But what is an endpoint or an outcome? Well, per the National Institute of Health, NIH, is just an endpoint is a targeted outcome of a clinical trial that is statistically analyzed to help determine the efficacy and safety of that therapy being studied. Again, I think a long-winded way for me of saying, what are you measuring, right? The endpoint, the outcome might be a change in height of a patient, right? Are we trying to help folks grow to a certain height, or weight, a change in weight? Do people on the drug lose more weight than people not on the drug? It could be heart attacks. Do people have fewer heart attacks on the drug than not on the drug? That when you think endpoint or you hear endpoint, don't think it, in some studies it is mortality. Do people survive longer? But that is not how you should think at large about endpoint. It shouldn't be a scary term. I like to just think outcome measure. And it can be there's other kinds like functional. You guys know the SARA scale possibly, or in FARS scale. There's biomarkers, wearables, imaging, blood tests. There's a lot of newer kind of outcome measures. Some certainly really cool stuff happening. But I also encourage you guys to go to, if you haven't, I know there's some resources I think on the NAF website that Celeste has been working on. Clinicaltrials.gov for folks here in the US, great resource. They put a lot of information on there. It's free. Anybody can access it. It can be a little, there are some nuances to using it. And I, again, encourage you to work with the NAF to find the right ways to work through this website. But it's a lot of information. Every drug company, as we do, companies or even academia, as we go into clinical studies, we need to put our information on this website so that people can see it. And often, and really what we're supposed to do also, and most companies are getting better at this, is putting your results on that website too, so that you should show your results if they answered the question and it was yes, or if they answered the question and the answer was no. All the results should go here, because that's how we will move science forward. We can't just talk about our wins and ignore the what we think were the no's or the failures, because again, they're not failures. They help answer the question and move science forward. And the only way we do that is if we can see everything. So I think drug companies are getting better. I know I'm very excited about my company. We're very positive on this, and drug companies at large are starting to share all of their information on this website. So I encourage you to go there. But here's an example from I think this is from Skyclaris of their outcome measure. One of their outcome measures from the MOXIe study was the change in baseline of peak work during exercise testing at week 12. So it was over 12 weeks. And it gets very complicated because there's different parts of studies. And I encourage you to take a look, but don't get overwhelmed if you look at these and you don't understand everything. But NAF again has great resources on many of this. It talks about describing that measure and the time frame. So the information is there, so we can all look at it and say, all right, what were also the inclusion and exclusion criteria are here. So all the information is on clinicaltrials.gov. Encourage you to go there. So that's a long-winded way of talking about endpoints, or what I think of as outcomes. So just what is the study measuring? How much does drug development cost? You may have heard it does cost a lot. Here's information from the CBO, the Congressional Budget Office. They put together that talks about the cost of a phase 1 study. And this is on average, right? Some studies are really expensive because they're really big, or studies have lots of different ways of measuring, and so that those can get expensive. Or some studies are smaller and cheaper. So this is just a total. 28 million for the phase 1. 65 million for phase 2 and 280 for phase 3, and from this work. You can see it gets more expensive in those later studies because typically have more people in the study. And what's interesting here is that this also talks about the cost of drugs that didn't make it. So you can see it's actually more for the average cost for the drug that was approved, 375, but there was on 690 million for drugs that didn't make it. And that's part of the business is working in pharma. We know that we may do a lot of work and it might not work, but it still answers the question. It's a very sad day when you see the results and they're not positive. I'm sure folks have seen some of that in the news on things, but it is moving science forward, and we recognize that cost. So some information from the CBO on that. But why might drugs fail? Or this is kind of how I would say, again, answer the question in a negative way. It could be for efficacy, could be that the drug didn't show an effect, or the effect wasn't enough, right? It was too small. It could be safety, that there were too many adverse events or side effects. In the pharma world, insider lingo, we typically call them adverse events, or they were too severe. The adverse events were just too severe for the benefits or the efficacy, the effect we're hoping to achieve from the drug. Or early on, this is interesting data that often it's for commercial reasons. And the way to think about that is that maybe there's just no advantage over standard of care. The drug has a small benefit maybe, but there's a great drug on the market already. So why are we going to bring a drug and expose people to more possible side effects? Because again, often there's benefits and risks. If there's already a drug on the market that works great, we shouldn't be bringing another drug out to the world. So over time, as you saw kind of that traditional view, again, those later phase 3 are more about efficacy. So drugs often fail for efficacy or don't have positive results in phase 3 because of efficacy, and safety a little earlier as well. So there's a lot of reasons drugs might not work, but again, every study is hopefully moving science forward and getting closer to medicines. What happens after a drug is approved? Well, that's when we have manufacturing plants that are people that have been waiting, ready to push the start button. Part of our filing with, say, the FDA about the efficacy and safety of the drug, also part of that filing is how do we make the drug? They do inspections of our plants to make sure that the drugs are made in a very controlled way, that the purity of the drug, the stability, how long can it sit on a shelf, like all of these things that we tested for the drug in clinical studies often can also match what's done in the real world. So we've done all that work. We have gone through all those inspections, and we're ready to start that manufacturing process and start shipping that drug out to the world. That starts after the drug is approved. Also, education and promotion, websites, getting the word out to patients and providers, healthcare providers. There's a lot going on in the world, so we have to make sure that when something is new, that people find out about it. We're competing with social media these days. We're competing with so many things. We want to make sure that especially healthcare providers early on are aware that PI, that prescribing information, what information is in that they can then make an informed decision, have all the information to say, okay, there's a drug now. I understand the benefits and risk. I understand the patients that were studied, that then they can possibly use it with their patients. But we don't stop the learning about the drug. We have phase 4 studies where we think about a different delivery device. If we had an IV, we can change the approach. Or if we have people have to prepare a needle and give themselves shots. Maybe there's an auto injector to give yourself a shot. Makes it a little bit easier, more convenience benefits to make it easier for somebody taking the drug. And think about, can we make the drug last longer? Can we reformulate the drug in a way that makes it more stable in the body for longer, so there's more time between doses? But also importantly, working on reimbursement. There certainly, as you saw those costs from before, there's a huge reimbursement aspect here. And the one point I would, from a pharma perspective, I would share, many times people hear about the price of drugs, and I don't have time to go into a presentation that could be a whole hour on its own. But when I think about the price of drugs, there is certainly a price to society. There's a price to individuals, but often, at least I'll speak about the US, there is a reimbursement process, either through the government, Medicare or Medicaid, or through private insurers that are taking care of much of that cost. Some of it can be passed on to the individual, but much of it, again, is through an insurer or a payer in the US. And something we, on our side, think a lot about, because we don't want anybody not to be able to get a drug. Often, many companies have access programs or ways to help with costs that anything that the patient would bear. for that drug. And I also say that, from a rare disease perspective, while drug prices can seem high, from a payer perspective, they're also often looking at, they're looking at all of their members. If you're Blue Cross Blue Shield, you're thinking about all of the people that you have under that you're insuring. And so, yeah, maybe it's one person that has a high cost drug, but that's really a small amount in total compared to all of the people that are maybe on diabetes drugs, cardiovascular. So while you may see those high prices for rare disease, it actually can lead to more investment, and maybe actually isn't as, for the payer perspective, is a little easier for them to think about, because again, it's about the total cost, not the cost for any one person. But again, that would be a topic for a huge day. So what kinds of jobs are at a pharma company? Clearly, there's research scientists, and if you're a research scientist on the phone, don't get offended by any of my depiction of you guys. Work you can imagine, working in the lab, developing the compound, doing the experiments to make sure the compound is safe, say in animals or in preclinical work. There's a huge amount of operations that goes into pharmaceutical development, making sure that all of the documentation is appropriate, making sure that we can make the drugs, making sure that we can get the drug out to people in a safe, if the drug has to be refrigerated, that can get out. Sales and marketing, we talked a little bit about, sorry, I'm starting to lose my voice. Manufacturing, can we make the drug in a safe and appropriate way? Can we make it at large volumes, not just sometimes it's easy to make things in a small volume, but can we make it at scale? Can we make it in a large volume? When we're thinking about protein therapeutics, as I talked about before, that is a very important consideration. Can I make it in a large bioreactor, we call them? It almost looks like, to me, a big tank of how they make beer. It's a big, huge tank, and they need to make these proteins. They do it in a very controlled process. The finance piece, very important to make sure that pharma that we can bring in enough revenue to actually keep making drugs. Human resources for these companies are typically not very small, so you need that HR component. Huge IT ask, especially nowadays with tons of data being generated. Can we use that data to our advantage to think about who's the drug working best for? How do we understand the genetic components of the disease? Medical directors, we have many in-house physicians that work to understand, the impact on, are we using the right measures? We understand that the clinical approach to the drug. They actually vet all of the clinical programs and sign up, says we think this is the right program for a trial for a patient. We also have reimbursement experts that talk to insurance companies to make sure whatever pricing we're thinking about is appropriate and will be something that can be used in the market. And I think that it's interesting to talk about is the hope of the pharmaceutical development cycle. It doesn't always work this way, but this is, I think, the way that at least I hope that it works, is that it often does, as I said, start with that discovery and preclinical science, which moves into clinical development, which hopefully commercializes a drug, then that we can get to an approved drug, which then we can commercialize. And a company that's in our shoes can get some of that, recoup some of that investment and get that return on investment. But really, a lot of that should be going back to early stage funding sources so that we can start the process over again. So we keep going around and keep developing new medicines, so that as a medicine gets approved and start to lead to sales of the medicine, that it can get to more medicines. Because if we just do one, I think we've failed. I think the idea of science is you never stop, you keep moving forward, and that for that to happen, this cycle has to keep happening. And I think a good example of that, as I said, I am from Georgia, and I'll say that don't take this as a dig at Coke. I love Coca-Cola, but I'll pick on them for a minute. Coca-Cola, again, love it. Don't tell my dentist I do drink it too much. But the main thing they've changed in about 100 years is the bottle. Despite a foray, if anybody is, I'm dating myself, but if anybody remembers New Coke, for the most part, it's in Coca-Cola Classic, the same formula for the most part for a very long time, with some bottle changes. Compare that to the drug industry, and I'll pick on Pfizer for a little bit. And sometimes they give them the press a lot for good or bad, but one of their products in 2003, Lipitor, they made a lot of money on Lipitor. Now, I think you can get Lipitor for about $3 for a month. It went off patent. It means that anybody it can go generic, that it can be made by lots of different companies now. There's no patent for Pfizer anymore on this product, and so it's very cheap now. A lot of the products that Pfizer have are very different now than they had even 20 years ago. That's their vaccine for COVID-19, an example of that. So it just shows that in the pharmaceutical industry, we're constantly reinventing ourselves, constantly trying to find new medicines to bring to help people living with diseases. Again, not a dig on Coke for my Georgia folks there in the audience, but just to show how we're constantly reinventing ourselves and finding hopefully ways to improve folks living with disease and hopefully to improve the world. I'm a little biased. I'm on the inside, but that is kind of how I think about it, and why, again, back to my why I want to work at a company that's developing medicines for people. And what does the future look like? I think the future is very exciting in the overall space, overall pharmaceutical development, but also in ataxias. The power of human genomics with the data we're getting out of the Human Genome Study and many other really, really cool genomics databases to really find what are maybe predictors of disease or what are targets at the DNA or RNA level we can target to change the course of disease. Very exciting stuff, especially in the rare disease community. Advances in measurement. You guys may have seen some work in wearables, gait monitors you wear on your ankle to see if your gait, the way you walk, has changed on a treatment versus not on the treatment. Some really cool stuff with some of our smartwatches or our phones that really get great data in a way that I think can really sometimes, maybe there's a lot of work to be done here, but maybe able to better understand the impact on somebody's life in a more meaningful way than in the time they're in a clinic, going through a certain test in the clinic. But also, as I mentioned earlier, innovations in study design. A lot of good work has been done in the cancer field. I think other fields are learning from those too, of how do we think about making shorter studies, studies with smaller people, not smaller people, studies with smaller numbers of people, studies that maybe compare not to a group of people not on the drug, but to the natural history of a disease. Because maybe we have good data to show how the disease normally progresses, and maybe we can compare to that. Really innovative thinking. Sometimes the FDA or other groups say, no, you're getting a little too cute, a little too novel of an approach. We like the old school approach, but certainly as a field, I think folks are moving ahead. And I don't want to pick on the FDA. They are a great partner and really are thinking how they're protecting patient safety and hopefully finding new, more efficacious drugs and thinking very creatively. Sometimes they're spurring us on and say, guys, you're being boring. Pharma, you need to think more creatively, think about new approaches, think about new study designs, new measurements, and really they're pushing us too. So we're all doing it together, advocacy, industry, regulatory bodies, and others, and really trying to move more medicines through the pipeline and hopefully get to more medicines in the real world. So with that, I saved a few minutes. Sorry, Celeste, if I went too long. Let's saved a few minutes for questions.
Thank you so much, David, for a fantastic talk. That was jam-packed full of information. We have a few moments for questions that have already come in. So our first question is around clinical testing cohorts. There's someone here who has read about how oftentimes there's more men than women in different trials, and people are usually of European descent, and they're wondering if including people from more diverse backgrounds would be a good thing, or if that would, you talked about variability in clinical trial groups, and that's why you need inclusion-exclusion criteria. Is more diversity a good thing? Could you speak a little bit about that?
Yeah, certainly. And that also is a topic could take a very long time. And it is something we think about a lot about diversity, clinical trial inclusiveness in clinical studies, because the thing we don't want to do is design a study that says, well, it worked great in this population, but in the real world, it doesn't work because people in the real world might be very different than that study population. And that's what you're really hoping to achieve is we can't test in everybody in the real world. That's not right for those people. We don't know if it's really a drug yet. So we want to make sure we have a representative, is what we say, representative sample, a representative group, cohort of people. I would say, historically, I think maybe compared to the way studies were run in the past, the drug industry has gotten a lot better. I think there's a lot of examples of where, there were more men than women in studies. You could even look at some of the early animal studies where I believe the mice are often all male mice instead of including female mice. And the way that men and women, the way our genes express different proteins can vary, right? Certainly. And so there's a lot of factors that the variability that goes in, and we do want to create a sample that has the right amount of variability so that if we get that answer, that hopefully that can be the same answer we would get if we looked at the full population. I think we're getting better at it. I think there's still room to go, but it is something that we work on very closely.
And our next question ties in with what you were sharing about clinical trial designs on one of your previous slides. we have someone here who has one of the ultra rare versions of ataxia, where you have a very limited number of people worldwide who have this condition. Could you speak a little bit about the different designs that are being used to develop pharmaceuticals for folks where the total population could be 100 people or less?
Certainly. So there's things that I think are, I don't want to say they're common, but more I see a lot more of, but we don't go in what we call healthy subjects or people without a disease or out of condition. We go right to people who have the disease for faster, but often maybe the way it works in those people could be a little bit, and you might combine that with an efficacy component, or a dose escalation or increasing dose component, and trying to think, how do we kind of smush the different parts of the study together? We might call that a 1B, 2A, which is really just a way of saying the part one, phase one, and the part of the phase two just kind of smushed together. So that's one way that folks, that there are many examples of studies like that where 1B, 2A. And maybe sometimes phase two studies can be what we'd say is registrational, where we can file with a regulatory body and we can register based on just maybe a phase two study for a rare disease. That there's ways again, the FDA is pushing forward on ways. You may have heard things like breakthrough and a lot of other terminology on ways to go faster and smaller on these studies. There's even we know some diseases are a very, very, very small amount of people. We call sometimes call those like N of one studies, and we're thinking, and this is a little more on a very innovative front, but there are some examples where a drug is made for one person, right? And again, very, very unusual, but I think in the industry we're thinking ahead to how do we reach more people, because we know that the days of just reaching just diabetes and cardiovascular disease, like that's not the future. There's a lot. Rare diseases are not that rare, right, when you add them up in total, but they each have their own considerations that we need to think carefully about. And can we one of the examples I showed from oncology is called a basket study, where you can combine different diseases into one. Is that appropriate? I'd say it gets really tricky. Some of the statisticians I've worked with, they lose some hair in this process, trying to figure out, can we get an answer that's representative for the larger population if we do some of these more novel things? But I think as a field, we're really pushing ahead and finding some new creative ways to get good answers.
Amazing. And we have time for one last question. We have someone here who's interested in how companies decide what diseases they're going to work on. How can rare disease patients and rare disease organizations figure out how to get attention to their particular condition?
That's a great question. Something that I've worked a lot in. And I'd say in today's day and age, it starts with information. Is the information readily available? And it's something that, I've talked to Celeste and others about of getting just, the information about ataxia. You guys have great information. So when I was on, the drug development side or working at a company working on ataxia research, right? Great resource to go to the NFA. Oh, where's some great papers? What's in the pipeline already? So that when we have discussions and say, all right, here, we have an approach. Maybe we have a genetic approach we think can work at the genetic level and treat diseases. Well, what diseases should we go after? Because I'll be honest, you want to do everything, but you make choices and you say, all right, well, what do we think we can show the effect of this approach in a safe way, in a relatively short way? Because sometimes some of these companies are, we don't have funding for 20 years. We might only have money to take us through for a few years. We have to think very carefully. But it gets back down to information, and the voice of the patient is so critical to make sure that measurement, the outcome, or that endpoint that we're looking at is something that captures the effect of the disease, the condition on people in a meaningful way that is representative of that. So I think that the voice of the patient, again, an area I think the FDA does a lot of great work in, of hearing from the voice of the patient, and the National Ataxia Foundation has done a great job really hearing from patients, making sure industry is hearing from patients, right? Industry hearing from patient advocacy. And the role of the clinician is often a part of that conversation, because again, it's all of those pieces working together to advance things. And I think so. If you reach out to somebody in the industry, I'm happy to chat with people. Again, specific medical advice, talk to your doctor, but I'm always happy to share more. And for me, that is what motivates me every day when I can go home and say, this is, I'm hopefully helping somebody's life get a little better out there that's living with a condition. So, and I think that happy to reach out to folks later. Thank you, Celeste. Thank you, everybody, for the time. Hope this was useful. And everybody have a great day.
Well, thank you so much, David, and thank you for everyone for attending. I hope you found something valuable at this from this session. Sorry that we couldn't get to more questions, but wherever you are in the world, hope you have a wonderful rest of your day. Good morning, good afternoon, good evening. Hopefully we'll see you at another webinar soon. Take care, everyone.